This was interesting. Then we decided to follow with another approach to see how general it is. Again, using the IntelliCage, Ewelina Knapska designed a new task in which now the liquid is just in one corner. Remember 2 bottles. But one bottle is sweet water, one bottle is plain water. Now over 3 days, we score how much they are able to distinguish, it's called discriminative appetitive learning, between those 2 bottles. It's different. This is spatial location, so it's kind of more special task. This is more discrimination, it's a bit different in terms of processing of information in the brain. And you see that the wild type, both copies of MMP9 gene intact, they learn very well, up to 100%, they prefer one bottle, the sweet bottle over the other one. Whereas the knockouts, both minus genes are disrupted, they have problems. They are barely over the chance level, which is 50% in this case. However, if we now put in one bottle quinine, which is a bitter solution, they avoid, they don't like bitter taste. And in a second bottle is just again plain water, they have no problems with learning. So again it shows that they are deficient in appetitive learning tasks, but they are perfectly normal in aversive learning tasks. To search for the neural structure involved, for some reasons I mentioned, we focus on the amygdala. Amygdala, this is how it looks in humans, in red here, is a part of the brain that is attributed with processing of emotions, storing emotional information, memorizing emotional information, and so on. It's actually quite complicated. In rats or mice, we distinguish over a dozen different subdivisions, different components, like shown here. And some years ago, we noticed that when we do this place preference versus place avoidance task, sweet water or air puff, sweet water corner or air puff corner, that when we looked for the CFOS expression, the one that we showed to be learning induced, and it is, normally in the brain, CFOS is almost missing. But after whatever we do to the animals that is novel, CFOS comes out. And it is expressed in the nuclei. So with specific antibody, labeled like peroxidase or whatever, we can see black dots, because they are just nuclei, which are round in shape, and only neuronal nuclei is present under those conditions. So we can actually score the number of these black dots, and each black dot reflects a single neuronal nucleus containing CFOS. And this is within the amygdala. This is called part of lateral, basal, and some other, and this is central amygdala. And you see here that under place preference conditions, central amygdala is kind of loaded with CFOS, whereas under avoidance conditions, CFOS is almost missing over there. So we decided to look what is going on with central amygdala in this context. So again, the same task. But now we look for MMP9 there. I remember that we believe that CFOS drives MMP9, so those are intimately linked. And we developed a novel system to interfere with MMP9 activity. This was done by Mayan Chaturvedi in the lab, who actually yesterday defended PhD thesis partially based on this work. Mayan produced nanoparticles, so-called P and J nanoparticles, that are about, 100 nanometer in diameter. This is the size of the lentivirus. That are loaded with recombinant, producing human cells, recombinant TIMP1 protein, that is slowly released from those nanoparticles over days. And these nanoparticles were injected just into the central amygdala. This recombinant protein contains so-called histag, that could be followed with immunofluorescence, so we know where is the protein. And then the animals were subjected to this discrimination task. As you see, just blocking the MMP activity in just very small part of the brain produce exactly the same effect as the global knockout of MMP9. on appetitive learning, and the same lack of effect on aversive learning, this queening versus plain water. I'm finding this fascinating, that first of all, a single protein is able to distinguish between such global dichotomy of learning, appetitive and aversive, and secondly, this protein operating in a tiny, tiny part of the brain, central amygdala, actually medial part of the central amygdala, produces or reproduces the same effect. We also know that MMP9 is there and active, because when we get the amygdala and look for the dysproglycan cleavage after exposure to sweet water, we see the cleavage. After air bath, we don't see the cleavage. And when the central amygdala is just taken away and the protein is isolated and dysproglycan tested, that sweetened water produces the cleavage and the air bath does not produce the cleavage. So during this training to sweet taste, there is MMP9 activity. Furthermore, one of the measures of, I mentioned that the plasticity is mostly equal to physiological properties, especially property called long-term potentiation, in which one can stimulate specific pathway, putting electrodes into specific axons that drive response in postsynaptic cells, and then record from these postsynaptic cells. And if especially organized stimulation is produced, especially high-frequency stimulation, so-called tetanic stimulation, what could be theta bursts or whatever, then one can recover the responses that are high. In this theta burst here, so this is 100%, so this is the basal responses, then this is this stimulation produced, and then you see that the response is like 150% of the original one. So there was a change, a plastic change in the connection between those axons and those responses. And you see that if we have the connection from basal amygdala to central amygdala, in the lacking MMP9 knockout, this increase lasts for about half an hour and then it's gone. And then to be sure that it's more general to just MMP9 knockout, but it's really MMP dependent, we use rat, not mice, slices of amygdala and use chemical inhibitor of MMP9. And the effect is the same. Also after half an hour, 40 minutes is gone. Interestingly, when we look at the NTP evoked by cortical input to so-called lateral amygdala, there is no difference in both cases. And this is well established, especially by Joe Ledoux and others, that the lateral amygdala seems to be critical for aversive learning. And aversive learning was intact. So it makes sense and looks more or less convincing. MMP9 could be involved in the plasticity, learning, and memory because it's located in the right place. And this right place is those dendritic spikes I mentioned that harbor excitatory synapses. We show it again at different levels, starting from here. This is the excitatory synapse. Those vesicles contain glutamate. We know that it's excitatory because at the electron microscopy level, excitatory synapses are asymmetric. They have strong labeling of so-called postsynaptic density here. Inhibitors are symmetric. And then, so we know that it's postsynaptic, and this is immunogold technique in which there is an antibody against MMP9 that is labeled with gold particle, which is like 10 nanometer in diameter. So that you can visualize the location of the protein targeted by this antibody by following this pattern. So it's postsynaptic. Here is the immunofluorescence at confocal level, in which we have antibody against MMP9 and look at the colocalization with NMD receptor, which is one of the glutamate receptors that decorates dendritic spikes. And it colocalizes. By the way, it does not colocalize MMP9 with GABA receptors, inhibitor ones. And then, this is the most important technique here, it's called in situ zymography. So it shows the enzymatic activity, zymography. And in situ, because of the slices, there's a compound called dicoin gelatin, which is gelatin, which is a substrate, I told you, of MMP9 and MP2, that is overloaded with fluorescent group, so much that they quench each other. It has to be cleaved in order to reveal fluorescence. So now we just look for the activity, not the protein, but the enzymatic function. And now in blue we have dendrite. MAP2 is a marker of dendrite. In red, in this case, we have NR1. This was what was green here. NMD receptor. receptor. You see the spines. And with high resolution at the spine level, we see MMP9 activity very often coinciding with dendritic spines. What is even more interesting, MMP9 is very low abundant protein and mRNA in the non-activated neurons, but after strong activation of their seizures, this increase. So this is after this activation, by the way. And also what is interesting is after the strong activation, not only protein and activity is located there at the dendrites and spines, but at the dendrites we also see mRNA. This is mRNA with autoradiography, so-called low resolution in situ hybridization autoradiography. The whiter signal shows MMP9 mRNA. These are the cell bodies in the hippocampus of the dentate gyrus and the cornu ammonis fields, CA1, CA3. After this strong excitation by giving a glutamate analog, so kinase, this is 24 hours, the neurons in the CA1 and CA3 start to die because they cannot survive so much excitation. But the dentate gyrus stays and survives pretty well. And you see that the signal now is not only in the cell bodies, which are here, but also in the dendrites of the gyrus. So somehow a message of mRNA moves toward dendrites after this kind of stimulation. This is important, because when we think about the neurons, we would like to have the synapses as a separate entities. If the synapses are supposed to delineate the connections that has to be maintained as stronger or weaker, they have to have a mechanism to keep. And biology says the only real mechanism to do something is always protein content. Yes? The proteins are carrying the function, or executing the function. So it's important to have proteins in specific location. So one of the ways neurons do is to have an ability to produce locally proteins in a specific synapse, not in the neighboring synapse. So in neurons, obviously the genes are transcribed to the nucleus, the RNA are produced, but many RNAs are not only translated into proteins in the cell body, but they could be specifically transported to a distant location, and then there's a phenomenon of so-called local translation. Interestingly, those RNAs, they are delivered there without poly-A tail. The RNAs that go into translation, they have to add a 3' end. Most of them, they have AAA, adenosine residues. And those they do not, so they have them locally first polyadenylated, and then associated with ribosomes, and then could be translated. So we're looking whether this phenomena may concern MMP9s. In this case, we use a very peculiar preparation called synaptoneuroscience, in which there's a special through centrifugation and filtration. There's a preparation that is kind of synapses, artificially kind of vesicles, big vesicles, that contain presynaptic site, postsynaptic sites, glutamate that could be released, postsynaptic density, and this kind of structures. They keep the biochemical machinery within them. So they can be stimulated, and like here with glutamate, and this is a measure of this poly-A ending. I told you that before translation they have to be polyadenylated. So those are this is MMP9 mRNA detected, and this diffuse signal means increasing number of A residues here, added at the 3'. So they are polyadenylated. They are also actively translated, because this is, in this case, is a separation from this. The preparation now is disrupted and ultracentrifuge to produce fraction in which we have RNA bound to different number of ribosomes. You know that if there is more ribosomes sitting on mRNA, it means that the mRNA is more actively translated. So there is a fraction with very little ribosomes, there is a fraction with more oligo-ribosomes and polyribosomal fractions. And you see that after stimulation, there is this again MMP9 mRNA detection. This moves to a fraction that contains more ribosomes. So it's more loaded with ribosomes, so we believe more translated. And indeed it is translated, because we can even detect the protein from this after the stimulation. Stimulated by glutamate, this is MMP9, and I told you that it's active and non-active form, the latent and cleaved. Both could be detected here. And this increase of MMP9 in this fraction is blocked by enzomycin. That is protein synthesis, protein translation.